welcome to the Industrial Biotechnology Innovation Catalyst webinar for February. If you haven't joined us before, these webinars run every month. They're a lunch and learn format for anyone interested in industrial biotech. Our upcoming ones are advertised on the University of Manchester's events page and also I think on the IBIC website. Sam will post a link in the chat so you can take a look. I think we're still finalising the titles for the March webinar. More about that later, but you can check back at the events page if you want to register and join. So for those who haven't met me before, I'm Sabina Hawthornthwaite. I'm the Programme Manager for the Industrial Biotech Innovation Catalyst, or IBIC as we call it. And our project is led by Aileen Miller and we're funded by the Engineering and Physical Sciences Research Council. They've given us just over £5 million for 4 years to try and translate research from the Northwest, from our universities into the Northwest industrial biotech sector. We're what's called a place-based impact acceleration account, and the aim is that we make an impact on the Northwest, so that we want to sort of supercharge the growth of the Northwest biotech region and deliver a low carbon economy. So we partner, the University of Manchester leads the project, partnered with Liverpool and also Manchester Metropolitan University, Salford University and Liverpool John Moores University. So over the last couple of years, we've had a wide variety of speakers at the webinar from our partnership and also from the wider industrial biotech community. And today I'm pleased to invite Professor Roberto Parra Saldivar, who's Chair of Sustainable Micro-Remediation at the Magan Centre for Applied Mycology at Cranfield University, alongside Dr. Aman Thakur, who's a postdoctoral research fellow in Sustainable Myco-Remediation, also at Cranfield. So Roberto and Aman are here to tell us today about the eco-innovation for planetary restoration, from regenerating land to myco-industrial revolution. So they're going to be covering, sorry, wrong slide. They're going to be covering how we can catalyse a greener, healthier and fairer future by transitioning from linear models of depletion to a circular loop of production and regeneration. They're also going to tell us about how fungal enzymes can serve as biofactories to drive the transition, and how we can heal soils devastated by extraction and warfare, transforming dead earth back into safe, productive land. So it sounds a very interesting presentation, and there'll be time at the end, as I mentioned before, to ask questions. Feel free to add them to the chat while people are speaking or raise your hand at the end. So I'll stop sharing now and hand over to you, Aman.
Thank you. Thank you, Sabine. Thank you for the great invitation. Thank you also to Professor Aileen Miller for this invitation, and of course the Industrial Biotechnology Innovation Catalyst. So this is a great opportunity to boost research, and for us it's a privilege to be here. So thank you very much. We're going to run this presentation with Dr. Aman and myself. He's going to start, so I will give the floor to Dr. Aman. Please.
Thank you, Professor Roberto. Thank you, everyone, for giving this unique opportunity for us to present our work, what we are carrying out over here at Cranfield. So the title of our presentation today is Eco-Innovation for Planetary Restoration, from regenerating land to a myco-industrial revolution. So we both, Professor Roberto and me, will be speaking on this. So a little bit of so a little bit of a background of Cranfield University, where we work. So Cranfield University works in different fields, different themes like aerospace, defense and security, energy and sustainability, environment and agri-food, transport systems, manufacturing and materials, as well as in waters. So where we sit beneath environment and agri-food, where this research England has funded a good project in the name of Megan Center of Applied Mycology that is dealing with different fungal aspects, and we are divided into different groups. For one, we are divided into sustainable food production, where mycotoxins and other fungal spoilages are being encountered with. Some of our groups are working in fungal biomaterials, where we are traditionally looking at in different packaging materials from the agro waste industries as well. So we are also doing work on fungal based bioenergy systems for production of fuels. We are also dealing with, we have a group with sustainable biomanufacturing that is working in upcycling materials with the use of fungi. And indoor, we're also looking at indoor and environmental mycology to improve comfort and energy efficiency in the indoor spaces. And our group, particularly the sustainable mycoremediation, is based in fungal based approaches for the eco-friendly bioremediation of the waste materials that are being produced. So we both are basically working in fungal carbon biosequestration. We are working in plastic biodegradation and industrial waste upcycling too from the waste materials. We are closely working in oil spillage, where fungi has an immense role, as well as in heavy metal remediation and hazardous pollutants. We will walk one by one in all these aspects to you, and also in the waste water mycofiltration units we are also working. So why fungi? Because fungi, as we know, that is in nature's master recycler. Nobody can come closer to a fungi to break down the toughest material, that is lignin. No other microorganism is there. And why it is so? Because it has such a powerful enzymes like lactases, manganese peroxidase, lignin peroxidase. So if you see the nature right now, so there is one fungi that is Armillaria ostacea, also known as the humongous fungus, that is covering an area roughly about 9 to 10 square meter and weighs approximately like 27,000 metric tons. So the mycelium, if with the passage of time, has evolved so much. And if we look at another example of Chernobyl, so the power of enzymes is shown in this particular fungi that is able to degrade, consume gamma radiation that no other particular solution is available right now. So this fungi is able to survive in these kind of harsh conditions too. So this given a very good example for us to approach that we followed in our sustainable remediation group to utilize both the mycelium and as well as the fungal enzymes. So to degrade most of the pollutants, most of the food material, so that a circular type of an economy can be constructed, and we can achieve a net zero approach. So in this micro-remediation, in this, why fungi is having? So we have listed some of enzymes, but these are not only the enzymes. There are different enzymes. It's an enzymatic powerhouse of producing enzymes. So it can produce lactases and lignins, manganese, glutinase, lipase, and other type of oxidoreductase enzymes that a bacteria itself cannot produce. So this is a powerhouse of production of enzymes, and if it can be translated, this can be translated, so we can utilize these enzymes as well as the mycelium culture to degrade lots of hydrocarbons, lots of agrochemical pollutants. We can also go with water and wastewater treatment. We have, in fact, it's there to degrade radioactive waste as well as, and also it can be there for the restoration of the mines and as well as the industrial effluent that is being there. So this fungi can help us to achieve all those things. And now the problem basically arises if we go dig deeper into this. So the problem is with the emerging pollutants like PH, polycyclic aromatic hydrocarbons from the oil spillages, POPs, persistent organic pollutants. Now with the regulations that are coming up against the PFAS and the microplastics. So in our wastewater streams. So there are different challenges, and we think that this fungal can solve most of these problems, and as well as also remediate other pollutants like pesticides. Our soils are being contaminated with that. Pharmaceuticals, after the COVID area, we are seeing a spike in these as well, and can improve and can provide a technology, because this is very important. The technology should be low impact, should be cost effective, should have a low impact on the environment, and can penetrate the complex substrates in this. This fungi has been traditionally also been very active in degrading like food waste. We know that utilizing food waste, there are immense lot of work being done in the production of mushrooms, in the production of mycoproteins, so that also can be utilized with fungi. That has not been well done with the bacteria at large. And it can also be able to degrade municipal solid waste and other agro-industrial residues. So to sum up everything that what we were talking before, that right now what we are doing, we are only taking the things from the earth, we are making out of it, and we are generating waste. And then this waste is again dumped upon the earth itself. There is no foolproof alternates to this. So what kind of an approach we should follow? We should always, yes, we need to take in order to survive. We should make, we should reuse, we should share, we should replenish, we should repair. But the most of the focus should be towards the recycling of this. And I think fungi is the one that can help us in recycling. So I give the floor to Professor Roberto to explain the different concepts in detail.
Thank you, Doctor. Thank you very much. So you see, this is a great opportunity to explore what nature offers, and we haven't tapped that much. For example, as Aman mentioned about the secretion of enzymes in all the realms, fungi are the best because it produces a really large amount of enzymes in order to survive. So we try to capitalize this power in here. For example, in the first pillar, we are going to basically we use a radical concept, try to use all the what we call waste and transform into feedstock for the transformation of waste into products. For that, for this reason, industry are coming quite a lot with us and try to find how can they use this waste and turn it into product. Because as Aman mentioned about the linearity, this is not going to be successful approach for the world. We need to find a way we can reuse, regenerate the planet. So for that, waste, I mean, we shouldn't use, or we shouldn't describe anything as a waste, but an opportunity to capitalize. So I'm going to talk about this terrestrial biomass, lignocellulosic, which is really staggering, 200 billion tons per annum of this waste, and some potential application also coming from the sea. Microalgae, for example, or from rivers, can be harnessed to take over all this runoff and transforming product. So this case of study is the lignocellulosic biorefinery. In this case, we study how can we use agro-industrial waste or agricultural waste that is really a lot of biomass. Think about for every kilogram of wheat, for example, we basically waste 9 kilos of bagasse or straw, and that is an opportunity for us to utilize. If we use a fraction of that, it will be a huge benefit for society, for the community. So what we done, this is what we we do in our labs, Aman and me, we use isolated of fungi that coming from all over the world, and then inoculate in Petri plates, expanded or growing in incubators, and scale it up in fermenters. And once we have this concept implemented, we try to use the extremophile enzymes taken from this fermentation into the field. So this is what we are doing. For example, this is an example of some of the work is CS43. It's an enzyme. It's a fungi that isolated from a mountain range in Mexico. This fungi, CS43, it's a fungi that produces a really large amount of enzyme. And we designed this very simple medium because we need to find solutions that can be applied in the real world without so many sophistication of cost. And this medium that we optimized here was based on this red bar that you see is the main component of this medium is tomato juice. So we can produce very good quality of enzyme using tomato juice, and we scale it up, as you see in the fermenters, and we got very good results. As you see, these graphics show the large concentration or the large activity of lactase produced under this media. And you look at the concentration at 25 liters, it reach up to 140,000 units per liter, which is really good activity to try some real world trials. So we've been working with this enzyme, and we are working in the degradation of plastics and degradation of some other pollutant, hydrocarbons, for the spillage of oil industry. And it works really well. And our idea, and actually is aligned with the mission of IBIT, is try to turn this into or translate it into a solution for the real world. So this is an example, and this is our roadmap of how we went for the large-scale production. First of all, we optimize the medium, and as I put there, the tomato juice, because we try to find a medium that is not expensive. It's really important to think about the large scale, because in the low scale, in lab scale, I mean, the medium, the 1 liter of media can be costly and it's not a problem. But when you scale it to the 1,000 or more, 10,000 liters fermented, then the cost becomes super significant. So this is the first pillar, I would say. The second is to try to find the reduction of time. So we need to make the process as fast as possible. One of the things that we need to take in consideration is fungi is a slow grower organism. Bacteria grow super fast. In 24 hours, maybe you have the whole kinetics ready. But in the case of fungi, it takes probably weeks. So our main concern in the optimization of the process was to reduce the time. So this is the second pillar that we consider. The activity, of course, we need to find an activity that is suitable for industrial application or environmental application. For that, thermostability, I would say, is the other pillar. And of course, then we need to think about the biotechnology advances that are needed and economical viability, because now we are in the last leg of the process, which is the industrial application. And we are working here in Cranfield at that leg. So we are ready to escalate the process and downstream enzymes that can be applied for the real scenario using these Pycnoporus sanguineus that produce these thermostable enzymes. This is a little bit of studies I'm presenting of this enzyme. This is a paper that we produced quite some time, but is the basic information that is required when you try to continue with the process that you believe is going to be suitable for scale up or industrial application. You see that the pH is quite. We have different graphics. For example, A, B, and C, and this is the pH related of two different isoenzymes. We have the Pignoporus sanguineus CS43 produce two different type of enzymes, lactase 1 and lactase 2. This is the way we call it. And you see regarding pH, they are quite similar, a little bit broader shoulder in lactase 1, but what was very important or very relevant for the process is if you see both enzyme has a kind of a plateau shape of thermostability that runs from 30 to 75 degrees, keeping almost 100% of the activity under these conditions. And this is a really good indication of our enzyme is suitable for industrial application, as the temperature will be something that we can not control, and maybe some impacted area, let's say the mining operation required the enzyme that can last at high temperature. But because the fungi was isolated from a very thermostable hot environment, semi-desert, these organisms evolved to produce a very robust structure of enzyme. So this is why we selected. Also, we try to put it to the level of test this enzyme. So we keep the temperature for many hours or as long as is possible to keep this experiment running to see how the thermostability will be little by little. Is a biological molecule that of course it will wear at some point. But one thing that was really surprising and we were very enthusiastic about the application is because the lactase, both lactase can last more than 1,000 hours at, let's say, at high temperature, 50, 60 degrees. It can last for so many hours. And that encourages us to continue this research and pursuing to use these enzymes for different applications. So that's the first pillar, the enzyme. This is another, well, the frontiers of fungi. The mission is how can we deploy the enzymes? We cannot use the enzyme just deploying the soil like normally comes out of the fermenter. We can talk a lot about this because, of course, we can produce the enzyme, but enzymes are expensive anyway in comparison with some other chemicals. Of course, are better, are eco-efficient, are not harmful for environment, for all the living organisms, but of course are expensive. How can we make this planetary restoration based on enzymes? Well, the avenue that we follow is try to use some platforms, so it's some support where the enzyme can last, and while the enzyme is still in the grass or in the soil, it will continue the effect as is thermostable and last very long time in the environment, whatever the conditions are. Of course, it will wear, but it takes a long time to lose the activity. Yeah. So we need to think about applied mycology experience. So again, the group here, we have a nice facility. I think is one of the unique groups that focus only in fungi, all the possible application of fungi. So we have this experience. We have this. Cranfield also is very well connected with industry. So that is why we also are bringing some industry connected to our projects. And of course, if we produce a novel enzyme that can solve this problem like plastics or pollutants, it become a niche area. So we can have the interest of companies because there is no many solutions, I think, for this matter. So again, the opportunity, of course, is sustainability, which is very important. Emerging pollutants has been the area that we were focused because, as I said, this is a good opportunity. Microplastics, PFAS, endocrine disruptors, and of course, fungicides, because also it's a problem. Pesticides, also we target with this, and also we collaborate. We are open to collaborate with everybody. That is the very, I think, best strategy to improve and to have something that is suitable, a global network. So this is a project that we are, let's say, evolving. This is a landfill. This is a mining field where it's eroded. You see, there is no plants at all. Of course, heavy metals are there. It's very difficult. We don't want to inherit this for the next generation. We need to find a way to neutralize metals. So for this, we are designing new approach. Like I will say, I don't have time to discuss everything, but it's a cocoon system which is designed engineerly, so it can restore the pollution in the mining, using, of course, as a core fungi for the degradation, enzymes that will be able to reduce the metals so we can have a safe environment, which is very difficult at the moment. This is what we know from industry. It's very difficult to regenerate these impacted soils because nothing can grow. So there is a need to find a way how can we restore. We are using fungi and the secretome of the fungi in order to reduce metals. So this is some of the work that we published and we worked before, the degradation of TNT. We're collaborating with many universities on these regards, on how can we treat explosives and some impacted molecules that are very high risk for the population. Of course, using enzymes, there is a paper there you can read about how we manage to degrade the explosive using lacases. And of course, the recalcitrant emerging contaminants, we can design. Of course, this is the next generation of treatment. We need to include the fungi. We don't know much about fungi. That's the reality. Most of the research is in bacteria, but there is a way, and you see there are some companies also that are looking at the PFAS degradation using enzymes. But of course, this is a big task that needs a lot of work and a lot of research. And microplastic, we are working on that also. We apply some funding on this regard using disruptive technology. How can we concentrate plastics and using microfiltration to breaking down some pharmaceutical and endocrine disruptors, bioactive molecules that can be degraded because, as you know, it is getting into the water, in the drinking water, high concentration of many pharmaceuticals and endocrine disruptors. This kind of given a dose in the water is something that we don't want. So we need to target also drinking water solutions. So for that, this is another area that is becoming very relevant, is the absorbent hygiene products, because you see the aging of the population require more these nappies. Also, the number of babies produce really staggering amount of these nappies. The solution should be something in the realm of engineering and biotechnology. So for that, we are working with enzymes that break the polymer. The polymers in it's a complex. The nappy is very complex. It has so many layers of different materials. So we are designing an engineering operation where we can use fungal enzymes to break polymers and cellulose and hemicellulose or cellulose system to upcycle and produce new value products. That is, of course, with the intervention of another colleague, Vinod Kumar, in this area, the upcycling. This is the structure of the two lag cases. If you wanted to know them, this is the picture of them. So these two enzymes are produced for CS43. You see very subtle difference between the two structures, give a big difference on the active site and therefore the substrate that they can take. This is another area that we are working basically with Professor Frederic Coulon on this regards on how can we treat PFAS and have a barrier, because it's not a simple molecule, it's very difficult to degrade this molecule. So we have membrane with immobilized enzymes as a first barrier, then biofilm that will take these byproducts, because the byproducts are going to be there. So this biofilm will be exposed to this molecule and cause a dynamic change of the profile of bacteria that are located in this support. And finally, maybe phytoremediation or degradation of the molecule at the end. Microplastic degradation, this is reports. I mean, everything of this, all this concentration of plastic found in humans, this is real case. Plastic is getting into our veins. One, we think we need to get rid of plastics, but there is a challenge of plastics because of microplastics in particular, not the large bags or PET bottles. The real danger comes with the microplastic because the accumulation of this is getting invisibly into our bodies. So we need to find a way to concentrate and break it with some enzymes. So for that, we are working in a system, but also we apply for funding. Hopefully we will get it. And the idea is maybe it can be a point of care technology in the future. So in mobilization, of course, you see, I just briefly introduced this thing. We need to find a way to immobilize the enzymes using magnetic carbon nanofibers. For example, if we wanted to remove in a lake, for example, biocompatible chitosan macrospheres and some other experiment that we done and successfully proved that it is possible to use these new technologies for encapsulation or immobilization to improve the degradation of these emerging pollutants. So this is the vision, how this micro-remediation will benefit the industry is by using these filters or households where we can have these filter systems where we can degrade these microplastics or emerging pollutant of pharmaceuticals. We mobilize, this is a work that we done with the Institute of European Center of Membranes. We mobilize enzymes into filter membranes for drinking water, and we found that the immobilized system allows the breaking down of the emerging pollutants, and then we can have safe water or safe even the heavy stream of concentrated product is not having these dangerous molecules, so it's not dangerous waste. So also magnetic nanobiotechnology parts or particles. We use this catalyst, based on magnetic particles or catalysts that we can separate from the system. We use biochar, and we are basically immobilize the enzyme that you see in the surface, and we use this catalyst to break down these molecules. Also the publication. This is a collaboration with St. Andrews University, so it was a really great experience. They have a lot of experience in the biochar. And with the enzyme, this combination gives us this nanobiocatalyst. In the case of this is probably another thing that we are also there because we are dealing with water. We work with water, so we were working in this project, but we got this experience probably to have a monitoring, a sentinel to evaluate the pollutants or the molecules that are produced in cities. We deploy this program at time of the COVID, of course, to assess the COVID impact and having a COVID zip code so we can trace up the building, where is the molecule. But water and the water monitoring is a really interesting opportunity, as you see here, to trace all the molecules and see what is happening in the city. That allow you to speed up all the information. Of course, this is sensitive information, so it is for the decision makers to apply some strategies. Yes, and this is the bioprocess. The last pillar is how can we get the idea, our idea in TRL1, going in the getting the funding to transform that idea into something that has the potential to be in the market, and then how can we design the process. This is very important, and I think this is where most of the research has some challenge, and I think EBIC is there, so it's really great to fill this gap, which is very important between the academia and industry, from lab to the global application. There's TRL 7 or 9, so basically we need to find a way, how can we escalate from maybe our Eppendorf into 300,000 liter fermenter, and what is the process? This roadmap is very important for all who wanted to start the business and you wanted to explore the potential to produce in large scale. From our perspective, which is bioprocessing, we assess the mass transfer, rheology of particularly mycelium, because we work with fungi, broad, the kinetic of modeling, and the scale it up, because it requires certain steps to scale it up. So that's the future. We need to explore the portfolio of new opportunities. With Aman, we work a lot of what is the waste and the composition of waste, and how this waste can be turned into product, nutraceutics, natural pigment, plastics or bioplastics. Bioplastic is not the same as the old plastic that has a lot of problem. We need to design and manufacture the plastic that is suitable for that. Also exploring new sources of proteins, as you see, is expanding the use of different insects, decayed plant matter, marine endophytes, and so on, to try to explore new products. Of course, using engineering, robust microbial consortium, not only the axenic culture, but also communities. It's important. And finally, we can improve and have a real process, scale it up. And a final statement, the future of industry is not gray, it is green, and it has some fungi there. It's not linear, as we discussed with Aman, it's circular, and it's not synthetic, it's biological. So together, I think we can build it. Thank you very much, and open for questions. I hope I didn't exceed the time. Thank you.
Many thanks, Roberto and Aman. So does anyone on the call have questions for either Roberto or Aman, either about the presentation or his work in general? Hi, we've got Neil. Hi, Neil.
Hi. Yeah, I can start us off. Hi there. Interesting. I got a question for you. I think it's around pillar 1. If I follow correctly, it was all native enzyme production. Is that correct?
That's correct. That's correct. Yes.
So, kind of two related questions. Do you know how the fungi regulates the expression and secretion of that lacase, and whether you need to change the culture conditions to maximize the expression of that? And I guess the sub part of that question is, do you have a feeling for the cost of goods for producing the enzyme versus, say, a kind of more classical recombinant protein production approach?
Very good, Neil. Thank you very much for your question. It's a very good question. And to be honest, we try the recombinant with yeast. I think it was Pastoris, Pichia pastoris. There is a challenge for genetically modified organisms to produce this level of lactases. And in this case, I believe the fungal secretory system is very difficult to replicate. Even cloning the enzyme in bacteria, because it will demand bacterial yeast a really high amount of energy and resources to keep coupling with this condition. How can we maximize? I didn't present those graphics, but we were working in optimization. First of all, we tried so many media coming from wheat bran and even other type of products. But after optimization, we got this media based in tomato juice, the best. It was the best, and it was, I think, science is also part of this luck. You know, we stumble in the right path using tomato juice. So we use tomato juice. That is one elicitor. That is one important step. Of course, we optimize several times the process. Also, copper sulfate is very important because the active site of the enzyme lactase, it has 3 copper sites. So you need to add copper into the medium. But of course, also you need to optimize because copper is toxic for the fungi. So you need to select exactly when you need to supply the copper and what is the maximum level that achieve maximum activity. So it's a combination of approaches and optimization that give us to this point. And as I mentioned, once you have a quite significant amount of lactase or lactase activity, then we started to explore the universe of opportunities because we produce enough lactase to try in the environment. This is the way we went, and once we reach this high level with this medium, we concentrate not in the production, but in the application.
Okay. I mean, is tomato juice cheap? It seems to have a very high water content. I don't know much about it, but so presumably it's quite expensive to ship.
Well, yes, the tomato juice it's a food, so it will be affordable, and it's not going to be 100% tomato juice. It needs to be diluted because act as an elicitor. I mean, a small concentration boosts the production. Of course, there is sugar and other components of the media, but at least this is it is cheaper than any chemical from Sigma or something like that. You can buy it in the supermarket and use it. Of course, need to be optimized again for each place where you're buying the tomato juice, but it's cheap. This is not expensive medium.
Okay, interesting. Thanks.
Thank you.
Thanks very much, both of you.
We have another question from Deepan Shah of CPI.
Yeah. Hi, yeah. So
Hi,
it's
Deepan.
very much related to exactly what Neil was asking about. You know, I'm from CPI, so I'm thinking about scalability, etc. And I mean, tomato juice seems reasonable. I mean, I think I was looking at bulk prices, and you can get tomato juice for $300 for 1,000 liters. So it doesn't seem like prohibitively expensive. I was just wondering whether you've done any techno-economics on it, and also the idea of circularity, where you're using something that's a food product as a feedstock, might not kind of feel right, you know. So it's just whether you've looked at the LCA around that. And my second question, again, very much related to what Neil was asking exactly, was that is considering producing it in, filamentous fungi like trichoderma, which are already used to very, very high level expression of cellulases and lipases and things like that, and using their regulatory pathways, just to make recombinant protein at very high level. And there you don't have to use tomato juice, and there are a lot of very, optimized media for those sorts of organisms. So I was wondering if you'd considered any of that. Thank you.
No, Deepak, thank you very much. Good question. I think that is something that definitely was to explore, using trichoderma, for example, as they have, that organism has that secretory system that we need to produce large amount of eggs. And so that will be great. I think that's a good idea. We can explore. Regarding the scale it up, we haven't done the techno-economical analysis or LCA. That's a great idea to explore that part, and we are going to consider to do that. I don't know, also open to collaborate. So the decision has been done basically on the scale it up on the substrate, which is cheap, and the concentration of lactases, because this is the main enzyme that produce this fungi. So definitely that's a good idea. Let's explore life cycle assessment and techno-economical analysis to take this process to the next level. Yes, thank you.
Thank you very much. Yeah. No, we're always open to collaboration, as I'm sure Neil is. So we should be talking. We get involved at a later stage than TRL 1, 2, 3, but again, there's a continuum that, so we should link up and
that's great.
Talk more often. Yeah.
Thank you.
Okay, thank you. So I just want to say thanks again to both Aman and to Roberto for a really interesting talk. Just to finish off, we've got another webinar coming up in March. We're going to have Dan Healy and Sahara Banot, who are the co-founders of Lanthogen Bio, coming to talk about their experiences as a new startup, and how they're working on their idea and looking at investment. Title to be confirmed yet, but we're working on that at the moment. Excuse me. And then the other thing just to mention is that we have a big pot of money through IBIC to help transform your ideas into impact and accelerate those ideas. So we have partnership development funding to make new relationships between academia and industry. We have funding for secondment exchanges, both academics into industry and vice versa, to provide knowledge exchange and develop ideas for commercialization. We've got funding for commercialization to take your idea into either a commercial entity or to license or to develop your market towards that. And we have innovation acceleration funds by way of proof of concept funding to help you ensure that your data is robust in terms of ideas that you have, either to collaborate with industry to get your research into their processes and products, or towards commercialization, and funding also to develop sort of specific networks in the industrial biotech sector in the Northwest. So if you're interested in learning more about those, please go to our website, and the team, fantastic team here, be happy to discuss with you. We have cut-off dates of March 12th coming up, and then another one in June, another in September and December. So yeah, still funding available. So I think we've come to the end of our time now. We're pretty much on 12:50, so that's pretty good. Before we close, I'd just like to thank again both Roberto and Aman for sharing their insights and knowledge on the myco-industrial revolution, and to everyone who's joined us today. And we hope to see you again in March. Thanks very much.
Thank you.
Thank you, Sabina. Thank you, everybody.
Thank you.
Thank you very much. Bye-bye.
Bye-bye.
Bye.